So that's how apoptosis is good for you. Okay. Next is Campbell chapter 12. Do we have any questions I can answer really quick? Or you want to put in the chat or raise your hand? Okay. I think we're good then. Okay.
I think you can remember apoptosis as a pop quiz.
As a pop quiz.
Yeah. Except that it's not osis, it's quiz.
Yeah. Okay.
I think you should it's like apop, like basically like the cell pops itself.
Yeah. Yeah.
Like pops its insides, or like.
Yeah. That's a good way to remember it. So sometimes the lysosomes inside the cell will basically release its digestive enzymes, and then the cell kind of will just pop inside. Yeah. Okay. Next is Campbell chapter 12, which is the cell cycle, the mitosis part of it. Oh my god, Anthony, you're here. Dude. Someone forgot to start the meeting. Yeah, okay. Okay, moving on. So a cell's DNA, or its genetic information, it's basically called its genome. And so the length of this DNA is very, very long. And this DNA, so it has to be efficiently packed into cells for all of it to fit. So this DNA is packed into chromosomes, which is made of chromatin. And the chromatin is like the material that makes up the chromosome. Yeah, so a lot of these terms must be remembered. So the chromatin consists of DNA wrapped around its protein, and then this chromatin folds up further into chromosomes. So during mitosis, a single chromatid will replicate into two sister chromatids. So here we see one singular chromosome, but before replication, it is only like half of it. So each of this is one chromatid. Okay. And then these sister chromatids are attached at the centromere, which is in the middle. The proteins, yes, the proteins are called histones. Good job. So during mitosis, these two sister chromatids are pulled apart from the centromere and then portioned into the two resulting daughter cells, as we'll talk about later. Under normal conditions, there are 46 chromosomes in your body or your somatic cells, and then we notate this as 2n. Half of these chromosomes will come from your mother and half from your father. In gametes or sex cells, there are 23 chromosomes, and when you have a kid, you will pass all of these chromosomes onto your kid cells, which ends up with 46 chromosomes, half from your partner. So out of these 23 chromosomes, one of them is a sex chromosome. So you'll only have X if you're a female and X or Y if you're a male. What's going on?
Wait, hold up.
Yeah.
Can I ask a question? So does that mean that you only have, if you're like a boy, then you only have one Y chromosome in each of your cells?
So in each of your body cells, your somatic cells, you'll have both one X and one Y. But the thing is, in your sex cells, the ones that you pass on to your kids, you'll have either an X or a Y. So you'll pass on either one.
Yeah, but didn't you say that there's like 46 chromosomes in somatic body cells?
Yeah.
Yeah, but
So
you said that
in your body cells, you
there's
have
one X. Is it like one X for one Y, or is it something?
so in your body cells, you have 46 chromosomes. Two of them will be sex cells. So sorry. In your body cells, you have 46 chromosomes, right? Two of those chromosomes will be sex chromosomes. One is passed from your mom, one is passed from your dad. So if you're biologically male, you'll have one X chromosome and one Y chromosome. Okay.
What are normal chromosomes called?
Normal chromosomes, they're called autosomes.
Okay.
Yeah.
Oh yeah, okay.
And then in your sex cells, however, since your chromosomes are basically divided independently assorted, you'll either pass on a Y or an X to your offspring. So that is randomly determined. Yeah. Okay. Oh my gosh. Okay. So mitosis, which we will be talking about, is the separation of these chromosomes. So cytokinesis is a little bit different, is the division of the actual cellular content that follows mitosis. So mitosis is the division of the genome, and then cytokinesis is the actual division of the cell. So the cell cycle includes mitosis as well as a time period between the divisions called the interphase. So during interphase, the cell mainly grows and develops, and the subphases in interphase are divided into G phases or growth phases, so G1 and G2, as well as S phases, so S for synthesis of DNA. And during the G1 phase, the cell experiences the fastest cell growth. So the cell basically obtains its nutrients and carries out its normal cell functions. And then during the S phase, the DNA is replicated or synthesized. So the DNA can be portioned into each daughter cell. So basically, during the S phase, one chromatid is replicated into two, and then so you have that. But it's not actually condensed into that form yet. You just have enough genetic content to form one of these. Okay. So then the cell will proceed from the S phase into the G2 phase, where the cell grows even more. And during the interphase, there will be many checkpoints, and we'll talk about those later. Yeah. Do you have a question, or are you just Marco? Okay. So after the G1 phase, there's a checkpoint in which certain cells can move on to a G0 phase where no replication happens and the cell just lives. We'll also talk about this later. And so after interphase, which takes up most of the cell cycle, is the mitotic or M phase, where the cellular content is divided with the genetic material. Okay, so mitosis actually has its own subphases. So the first phase is prophase. So during this phase, the chromatin will condense and become visible under the microscope. So before, the chromatin is uncondensed and you can't really see it, but then they condense into these chromosomes. And so these are arranged into sister chromatids, and the nucleoli, which is in the center of the nucleus, will disappear. Also, a mitotic spindle starts to form and assemble from centrosomes, which are responsible for forming microtubules and microfilaments. They will start to form, and the mitotic spindle, the term, consists of the centrosome and microtubules. So then the second phase is prometaphase, which is between prophase and metaphase. And during this time, the nuclear envelope will start to fragment and disappear, and the microtubules here will start to invade the nuclear area. And kinetochores, which are little hooks basically on each sister chromatid, will start to form at the centromeres. So these kinetochores basically provide a spot for these microtubules to attach on and pull the sister chromatids apart. Okay. So next, in metaphase, the chromosomes are pulled by the microtubules into the middle of the cell, which is called the metaphase plate. The kinetochore microtubules are now all attached to the sister chromatids. And so basically you'll have approximately one sister chromatid on each side, and then they will normally be pulled apart. So during anaphase, the cohesin proteins that hold the sister chromatids together are cleaved, and then the sister chromatids are pulled apart by the microtubules. So while there are non-kinetochore microtubules, so they're basically not attached to any of the chromatids, they will elongate. And then the kinetochore microtubules that are attached to a chromatid will shorten. And so that will pull the chromatids apart to the two different poles. And in doing so, the cell also elongates. So the cell basically like go from a circle to more of an oval shape. Next, during telophase, the two new cells basically form inside of one big cell. So the nuclei and the nucleoli will reappear, the nuclear membrane will regenerate, and then the chromosomes will decondense. And then lastly, in cytokinesis, it's the physical division of cells. So the cellular content is sorted, and then the cells are physically divided. So during cell division, cytokinesis will occur by cleavage. So basically, a cleavage furrow forms to split the cells. In this process, there are microfilaments inside the cytoplasmic sides, and then these microfilaments will work together to contract, and then that causes constriction at the center of the cell, and then the cell will eventually pull itself apart. Okay, so this process is a little different for plant cells, since in plant cells they don't have a flexible cell membrane. Well, they do, but on the outside they have a rigid cell wall that cannot just constrict and pull itself apart. So what they do is they have vesicles that contain of cell wall materials inside the cells, and then these will form a cell plate as the vesicles aggregate. So this basically forms a new cell wall from the inside out, and then that divides the parent cell into two smaller daughter cells. And so that inside will become the new cell wall of the two cells. So this process is much more simplified for bacteria. So they undergo binary fission, which basically it just involves the replication of genetic material followed by the cytokinesis of the cell. So there are two origins of replication where your DNA basically just like I guess like peels itself in half and then replicates on the two sides, and then that creates extra genetic material for the two bacterial cells to have. And they also eventually replicate by cleavage, where the two cells basically just cleave in. Okay. So to ensure the cell cycle doesn't keep the cells replicating for too long and cause cancer, or when the cells are damaged, these cell cycle regulators will help keep the cells in check. So one type of regulating molecule is called cyclin-dependent kinase, or CDKs. These kinases have to be attached to cyclin to function, but they're always present. So a very good example of a CDK is MPF, or it's called the maturation promoting factor, but it can be thought of as the M phase promoting factor. So it basically allows the cells to, it checks the cells for DNA or extracellular damage. Hold on, I'll finish explaining this, and then it will allow cells to proceed into the mitotic phase after interphase. Okay, I'll go back to the previous slide really quick. Okay. Are you taking notes or? Okay. I mean, these slides are a good addition, but please remember to actually study Campbell, because it talks about much more than I can cover in an hour. Yeah, okay. Cool. Okay, anyways. During interphase and also as the cell finishes interphase, there are other cell cycle checkpoints that can stop the cell if there is excessive damage to the DNA or the cell. So between G1 and S phase, there's a checkpoint that can move the cell to the G0 or non-dividing phase, if it's appropriate or if the cell is too damaged. So if the cell has excessive DNA damage, for example, the cell will be moved to the G0 phase to how do you say this? to basically fix the DNA damage before the DNA can replicate. So that ensures the DNA is not replicated in its damaged form. Also, certain cells, such as neurons, will be in the G0 phase for much of their life. So a lot of your neurons will divide when you're very, very young, but then once you get to be an adult, these neurons basically stop dying. Stop dividing. And so if your neurons die, then you don't really have replacement neurons. So also before M phase, there is another checkpoint to ensure the cell can proceed. So furthermore, signaling molecules outside of the cell, like growth factors, can promote cell growth and division. However, when the cells grow together and become too clustered, there's a thing called density-dependent inhibition, and then this limits cell growth to prevent excessive competition for resources, and the excess cells will start to die off. So failures in regulation of the cell cycle can cause cancer, which is uncontrolled cell division. Sometimes it is the DNA that is damaged, and then that results in a damaged cell cycle regulation protein that basically fails to regulate the cell cycle and division, and also the cell checkpoints might miss a couple of cells. So there are two types of cancer tumors. One is a benign tumor so those don't metastasize or move around, and they don't cause very serious they don't cause extremely serious problems. So of course, cancer is not good in any sense, but these are a little bit more controllable in that they can be removed by surgery or radiation, and they won't spread to other parts of your body. However, malignant tumors are very, very bad because they can spread to new tissues and are very serious, and then they can reoccur months or years after the initial tumor is removed. So, for example, a malignant tumor can, for example, move from the bone to the pancreas, which gets very, very dangerous because pancreatic cancer cannot be treated easily. Yes. What Anthony said. Okay. Next is Campbell, chapter 13. Do we have any questions? Okay. This is our last chapter for today, so we're almost done. Okay. So, during inheritance for offspring, the genes of the baby are always inherited through the gametes, which are the sex cells, the eggs, and the sperm, and never ever body cells or somatic cells. So there are two types of reproduction. In asexual reproduction, the organism basically makes genetically identical copies of itself. And this is seen in bacteria, where the bacteria just undergoes binary fission and becomes two different organisms. In sexual reproduction, the gametes of two parents are combined to create a genetically unique offspring. So this happens in animals like humans and can also happen in plants. So meiosis is basically the process for creating new gametes. So basically, regenerating your cells that will be merged together to like form a new offspring. So as we have said previously, in each human body or somatic cell, there are 46 chromosomes that include 2 sex chromosomes. In gametes, though, there are only 23 chromosomes that include 1 sex chromosome, since the 2 gametes must combine to form an organism. So when the 2 chromosomes from the 2 gametes combine in a new organism, the resulting chromosome pair is called the homologous chromosome. So each of these, so this is one pair of homologous chromosomes. Each of these is one chromosome from one parent, and each chromosome contains of 2 sister chromatids that are replicated. So so you will have, for example, a homologous chromosome pair of, let's say, chromosome 1, and then one chromosome will be from your mom and a chromosome will be from your dad. So these homologous chromosomes encode for the same protein types but can have slight variations in the loci or the location of the gene. So, for example, the loci for eye color is at the same spot on homologous chromosomes, but one can code for brown eyes while the other can code for blue eyes. So these homologous chromosomes that we're talking about right now are autosomes, and these are not sex chromosomes. The sex chromosomes, however, don't really follow a homologous chromosome pairing. So the combination of X and Y chromosomes will determine the sex of the offspring. So if you have two X chromosomes, so if one parent passes on an X chromosome and the other parent also passes on an X chromosome, the zygote will then become a biologically female offspring.
Oh wait, just a question.
Yeah.
If two trans people like make a baby, will the baby certainly be trans, or will it have a chance not to be trans?
And Aston, trans means that you're changing your legal gender. That's not like changing your biological.
Anthony,
Oh, okay, never mind,
no,
never mind.
Anthony, that's not a weird question.
Sorry, sorry.
No, I'm not
Sorry,
talking
I
about
get
you.
no. I'm just telling Marco.
Yeah.
Marco, like I'm sorry, I got it wrong. Sorry.
It's okay. It's okay.
Got my question wrong.
So trans people, the gametes that they produce will still be like adherent to their biological gender. So yeah. Yeah. Stop calling people's question weird, bro. Okay. Yeah. Okay. Yes, that is the type of fertilization that they can do. So also this is sort of relevant for like same-sex couples that can also take their own gametes and then do artificial fertilization and then create a kid. Yeah. Oh my God. Okay, guys. Yeah. What? Anthony, you don't know? So like you can take your gametes and like so a lot of people like preserve their eggs and then so for people with like fertility issues as well, they can preserve their eggs or their sperm and then try to like I guess like artificially fertilize them to see which zygote is the most viable and then reimplant it into convert gam. You're not converting gametes. Yeah, okay. Okay. Um so the number of chromosomes in each cell will determine the type of cell it is. So haploid cells, which are your gametes, only have one set of chromosomes and yeah. But two haploid gametes will combine into a diploid cell during fertilization. So diploid cells have two sets of chromosomes, which are the body's somatic cells. Oh my God. No, Joy. No, don't think about what a dog and a human breeded would look like. Yeah. Joy, you're a little messed up. Yeah.
I agree. That's missed.
I'll agree that Joy's very, very.
Okay. Let's move on. Okay. So during meiosis, a diploid parent cell is divided into haploid cells. So meiosis is very similar to mitosis, but there are two phases, which are meiosis 1 and meiosis 2. So basically the whole process will repeat itself 2 times. Also, the daughter cells that come out of this process are haploid with only one set of chromosomes instead of 2 sets. So during meiosis 1, basically the homologous chromosomes, so one set from your mom and one set of your dad, they will replicate before they are separated. So one chromosome becomes two, as you can see here. And then before this separation, however, there is crossing over between the two inner chromatids of these homologous chromosomes as they line up in the middle. So essentially the genetic material between the two loci of the two homologous chromosomes are exchanged, and then this helps create more variation in the produced gametes. So as you can see, and then during meiosis 2, these chromatids are split further. So if there is genetic material exchange between the two middle chromatids, these two chromatids here will also be a little genetically different than the two outer chromatids. So during meiosis 2, basically the sister chromatids are separated, and that process is basically the same as mitosis. So all of these phases have prophase, prometaphase, metaphase, anaphase, telophase, and cytokinesis. However, they're basically just numbered by whichever process they belong to. So like prophase 1 and prophase 2, that's basically what differentiates them. And also remember, during meiosis 1, the homologous chromosomes are separated, and then during meiosis 2, the sister chromatids are separated. Guys, stop talking in the chat, please. Thank you. Okay. And then meiosis also, as I said, it helps to create genetic diversity. So the first way is through independent assortment. So during meiosis, when the two homologous chromosomes when they line up, they will line up in random assortments. And so this allows for random assortment of these chromosomes into the daughter cells. And the same goes for meiosis 2, when the sister chromatids are assorted into the gametes randomly. So as you can see here, the homologous chromosomes can either line up this way or that way, and then that will create differences in possibility of how they will be separated. So the second way is crossing over, which is seen in meiosis 1. So during crossing over, the material between the two inner chromatids will be exchanged, and then this creates recombinants that have slightly different genetic combinations that are passed on to offspring. So lastly, random fertilization will ensure that the fertilization is by chance. So basically random sperm with unique genetic material fertilizing essentially random eggs. Yeah, so these will help create genetic diversity and variation in your offspring. Okay. Oh, and that is also why you and your siblings are not exactly the same. But if you have twins that come from the same egg, they will look very, very, very similar because they basically have partially the same genetic material. Okay, that is my content for today. Sorry, I don't know why my screen keeps shrinking. My bad.
Wait, I actually do have one question, even though I just said no in the chat.
Yeah. What's your question?
So basically, like in the graph, it says those were like meiosis, right? Meiosis 1 and meiosis 2. And then like did it only show X chromosomes? Or like what do Y chromosomes look
So
like?
that's actually the arrangement of the chromatids. So they tend to arrange themselves in an X shape. So those didn't show X and Y chromosomes. Those just showed like general chromosomes. So chromosomes will like often line up in that type of shape. Hold on, I'll pull up an image for you guys. Okay. So if you can see my screen here, so each chromosome basically
Wow.
looks like this, and then after replication, they'll just like arrange themselves in sort of an X position. So they'll
Yeah.
just generally look like X's.
So this will be useful on who is the father, right?
Yeah, but then how do Y chromosomes form?
How?
Like I know that these are just general chromosomes. But then how do X and Y chromosomes form? Because like
I'm
the
not
Y
sure on
chromosome,
how they form. I just know like the shape is different. So that's why they call them that. Okay,
okay.
I'll look into it when you guys answer some questions though. Yeah. Okay. Anything else? If not, I'm gonna go ahead and ask you some questions that you guys will answer. And then I'll also look up Marco's question. Okay. Also wait, I saw someone else's question. So, fraternal twins are just from different eggs. Yes, fraternal twins are when two different sperm fertilize two different eggs. So sometimes multiple eggs will be released. That's very, very uncommon. I'm not exactly sure on how that happens, but yeah. Okay. Questions. Okay. Okay. So, your first question, is that the same for identical? No. Identical is the same egg. Yeah. Okay, your first question. Order these phases. W, metaphase, X, prophase, Y, telophase, Z, cytokinesis. You can just message me.
Wait, can you repeat again?
Yeah, I'll repeat it once. So order these cell cycle phases. W, metaphase, X, prophase, Y, telophase, Z, cytokinesis.
Can you type like the processes like in the meeting group chat?
I'd rather not type them because it's viable is verbal, right?
Competition, it's not gonna be like that, but like I should be
Didn't you
saying
join last? You
yeah,
participated in this.
yeah, and then I just lost every single
That's just like Anthony, honestly.
Wait. Oh, so Derek. Okay. Okay. So the correct answer is X prophase, W metaphase, Y telophase, and Z cytokinesis. So during prophase, basically your chromosomes will condense, and then during metaphase they will already be lined up on the metaphase plate. During telophase, everything's already portioned out, and then during cytokinesis, the cells physically will divide themselves. Okay. Okay. This is just a general question, but what is the difference between plant cell cytokinesis and animal cell cytokinesis? If anyone wants to raise their hand to explain, you can also do that. Okay, Marco.
So basically, cytokinesis in animal cells, because animal cells don't have that plant wall, that pesky plant wall. Basically, they just like them. It goes through a normal process, and then it splits. The spindles form. The nuclear membrane basically vanishes, and then they split. The nucleus reforms, and then they just and then they just like divide. And then for the cell wall pesky problem. They vesicles basically they group in the middle of the cell, containing like the cell wall materials. I'm assuming it's like cell, and then like they just form a new cell wall, and then yeah,
Mhm.
basically that splits it. A
Yeah.
wall. Yeah.
Yeah, good job. Okay, that was such a good explanation. I'd appreciate if you participated like him. Be like him. Okay. Um, okay. So, okay, this is my next question. If a drug inhibited DNA synthesis, which phase of the cell cycle would the cell stop at? A, M phase, B, G0 phase, C, S phase, or D, G2 phase? Okay. If a drug inhibited DNA synthesis, which phase of the cell cycle would the cell stop at? A, M phase, B, G0 phase, C, S phase, D, G2 phase. Okay. So the answer is S phase. Because during S phase your DNA is synthesized, so they're replicated, which is what they call synthesis, so that's the synthesis phase. So the cell cycle would basically just stop there. And then the cell would not be able to replicate its DNA and wouldn't be able to move on. So the M phase is when the cells are already like dividing. So that's not the answer. G0 phase is when the cell will just G0, it's like a state of like just existence for the cell, so that wouldn't be where the cell would stop at either. And then the G2 phase, the DNA is already synthesized, so it wouldn't just stop at the G2 phase. So see, and DNA synthesis is the most relevant to the S phase. Okay.
I think that G2 was after like S phase.
Yeah, G2 is after S phase. Okay. It is 8 p.m. Thank you for coming. Sorry about the delay today. We'll try to be more on time next time. Yeah, Anthony. Okay. Good work, everyone. Thank you for coming. And see you next week.
Thank you.